Good morning, everyone, and welcome to this ACUR at ANU student presentation session. I'd firstly like to acknowledge and celebrate the first Australians on whose traditional lands we meet and pay our respect to the elders, both past and present. My name is Max Kirkby and I will be chairing today's session. Our first presenter is Dejun Cai. Jack, please start when you are ready.
Yep, so yeah, just start to share my screen. Can everyone see my screen?
Yep.
So, okay. Hi, everyone. This is Jack from the ANU. In this presentation, I'm going to give an overview of my research project, model calibration of a satellite data assimilation system for soil moisture prediction using Markov chain Monte Carlo algorithm, or the MCMC algorithm. The presentation is structured as follows. Firstly, I will provide some background information on the soil moisture estimation, including the concept of data assimilation and the calibration. Then I will outline the key objectives of the study and the methodology. This will be followed by some key results from model calibration and a summary of the main findings. The estimation of soil moisture is an important component in the water resource management, agriculture, and weather forecast. There are two major methods in practice to estimate the soil moisture: the models and the satellite data. The hydrological models can provide continuous prediction at different scales, but they may not be suitable for all areas. In comparison, the satellite data can provide objective information on the soil moisture with a global spatial coverage, but they have large areas in coastal areas and forests. So people bring this question: can we integrate the model and data in some ways? A new system called GRAPS is trying to achieve this model data integration by a method called data assimilation. It integrates the satellite data from the soil moisture active passive, or SMAP, into the antecedent precipitation index model, or the API model. The most important part of this model is the coefficient phi-t, which represents how much moisture from yesterday is still in the soil today, or the so-called the moisture memory. It is controlled by two parameters, beta and delta, which characterize the model behavior. And the data assimilation essentially adjusts the model output, which is the red curve here, towards these points of satellite data, which updates the model estimate to be more accurate. However, one problem of this system is that the beta and delta are treated as constant all the time, and this cannot capture the different dynamics of moisture at each area, driven by different climate conditions or the soil properties. To account for such spatial variations, people often rely on another process called calibration, which is to modify the value of parameters to help the model represent the behavior of the observed data more realistically. So apart from data assimilation, the SMAP satellite data may also be used as the reference data for calibration, which can incorporate such spatial variability and provides a new perspective to improve the model performance. We choose Markov chain Monte Carlo, or the MCMC, for the calibration. The main feature of this statistical algorithm is that it treats each parameter as random and try to get a distribution of different reasonable values to show our uncertainty. The Metropolis Hastings algorithm is a flexible MCMC method, and we use that for this research. So therefore, based on previous background information, our study has the following two objectives. Firstly, we will apply the MCMC method to calibrate the parameters in the API model against the satellite data to explore the spatial variation in the parameter values. Secondly, we will compare the model performance under MCMC calibration and the data assimilation to evaluate their effectiveness in the soil moisture estimation. We first test the MCMC calibration at 13 study sites in Australia, and then applied that over every 10 by 10 kilometers grid cell across Australia on the supercomputer Graphical. In particular, these 13 sites represent the Cosmos network, which is the first network of cosmic ray probes in Australia to provide ground-based measurements of the soil moisture. The model performance is evaluated by the correlation with these ground measurements from July 2015 to June 2020. As mentioned before, by running the MCMC calibration, we can get the distribution of beta and delta at each site. The peaks of this distribution, which are these red vertical lines, represent the most likely values. And we compare them across Australia to explore the spatial variations in the parameters, which is our first objective. In addition, this distribution allows us to randomly sample different beta and delta to run a model. And this gives a collection of different possible model outputs, or so-called the ensemble. We evaluate the performance of individual model run and pick the best one to compare with the result from data assimilation. Now I will start presenting the key findings from such MCMC calibration. Firstly, the value of beta after the calibration shows some interesting spatial pattern. The beta has high value in Northern Australia, which are represented by these darker green colors. But then the values start to decrease towards the drier Central Australia, and then back up again in the coastal areas in the southeast and the southwest. This spatial pattern is supported by the plot here, as beta has a non-linear but positive association with the wetness of the climate in each area. In particular, these patterns actually match the physical meanings of beta in the API model. We know that if the climate is wet, the soil can keep more moisture from yesterday. That means a higher moisture memory, and a large beta can give a larger value of phi in the model to capture this physical phenomenon. For delta, the values are quite small in the Northern Australia, but becomes higher towards the south. Based on this spatial pattern, we fit the model of delta against the ratio between the average summer rainfall and the winter rainfall. And you can see that there's a very consistent curve and a negative relationship. For example, for Northern Australia has a tropical monsoon climate with wet summers and dry winters, and the value of delta tend to be very small, which is circled here. So in summary, the spatial variation of delta can be linked to the seasonal rainfall patterns in each area. Next, the model performance shows significant improvement after such MCMC calibration. The correlation with the ground data is improved for 11 of the 13 sites. In particular, for Talagran in Tasmania, the MCMC calibration improves the correlation by 106% from 0.38 to 0.78, which is quite dramatic. This result highlights the benefits of MCMC to get the distribution of the parameters and then pick the one that can best represent the actual behavior of the soil moisture. We then compare with the result from data assimilation, which are these red triangles. In general, the data assimilation improves the model performance more effectively. This advantage becomes more evident when the original raw model already performs quite well, and the calibration didn't make much difference. However, the MCMC calibration sometimes performs better. For Robson in Queensland, the data assimilation improves the correlation from 0.39 to 0.59, while the MCMC calibration gives a slightly higher correlation of 0.64. We can see that these two methods show more or less advantage in different cases, but both of them can improve the model. So we may have the following question: can we get an even more accurate soil moisture estimation if we can combine these two methods? In fact, there's a very interesting graphical teaser for this combination in the Nangara in Western Australia. Both methods improve the performance by over 50% here. The graph on top shows the model output. The MCMC calibration, which is the red line here, can help the model pick up the systematic increasing trend in the ground data, which is the graph on the bottom. However, the outputs from data assimilation, which is the red line, can help the model better capture the random variations. This graphical observation showed that these two methods can actually refine the model estimate in quite different ways. So in the future, we may calibrate the model by the MCMC first, and then apply the data assimilation to further adjust the model estimate. This can probably complement the benefits of these two methods, and eventually push the boundary of the model accuracy for soil moisture estimation. So in summary, the site-specific MCMC calibration of the model can uncover some important spatial variations in the parameter values. In particular, the behavior of such spatial variation can be linked into the climate variables like the wetness and the seasonal rainfall patterns. The MCMC calibration and the data assimilation can greatly improve the model performance in quite different ways. That means they may complement each other, and integrating these two methods has a strong potential to produce cutting-edge soil moisture estimation, which will eventually support different hydrological, agricultural, and meteorological applications. That's the end of my talk. Thanks for listening.
Thanks very much, Jack. There are now 5 minutes for Q&A from the judges. Judges, do you have any questions for Jack? Yeah, feel free to unmute yourself and ask away.
Hey, Jack, that was really awesome. Thank you for kicking us off on a Friday morning with a really compelling paper. I mean, I guess my question kind of goes to the questions that you were raising at the end. Where does the research go from here? What's the next step?
So this research is actually a long-term project of the soil moisture estimation funded by the Bureau of Meteorology. So the system called graphs, which I mentioned previously, is like, so the initial idea is to basically achieve this model data integration. But then when I did the research, I found that I found a gap of this like a constant parameter issue where you know, in Australia we have very different climate conditions, but somehow the parameters are constant across Australia. So in the future, the research will be to find the appropriate method to efficiently calibrate the model across Australia, and then apply the data assimilation to improve the model performance. And that will be, then the next step will be just to run the model to see how it goes in the next 1 or 2 years, because the satellite data has only started in 2015. So the actual data period is actually pretty limited. But now we are still looking at some, you know, synthetic experiments, and then try to apply that in a real practice to see how accurate it would be. Yep. Hope this answers the question.
I've got a question, Jack. Well done this morning. It's always hard to be the first cab off the rank, so good job in getting in there. It was a really inspiring presentation. I know in your very last point you touched on you know, some of why this is important, hydrological, those sorts of things. In a practical sense, where does this go? How is this applied in the, you know, to generate real benefit or otherwise in the agricultural field? I guess what's the so what behind the research?
So you actually touched on a very good point, because this project is actually inspired by the needs from the farmers, because they try to get a good sense of the soil moisture before they start to plan the irrigation for the year. Because sometimes they, you know, if they, for example, they underestimate the moisture in the soil, then they will basically oversupply the water to the crops, and this will lead to the issue of waterlogging, where the crop just get too much water and there's not much oxygen left. So a better, I guess, but the previous research gap is that the estimation of soil moisture only have some small, like what we call the ground-based measurement in very small footprint. But now if we can really convey the satellite data across Australia, then we can basically get 5 by 5 or even 1 by 1 kilometers accuracy across Australia, where you can just pick a location and then I will get the estimate for you, and you can get a good sense of how much moisture is there in the soil and how much water you will apply in the irrigation. So yeah, so this is I guess this is the main part of the agricultural application. And I guess one of my research which touch on the spatial variability across Australia is that we're trying to incorporate some supplementary information about the site-specific condition at each site, so people can relate the model result, the parameter result, to their, for example, the climate condition or soil properties, so they can get the holistic view of how the dynamic will work and how we can fit that dynamic into our future agriculture planning. So yeah.
If there are no other questions from the judges, are there any questions from the audience? And if so, just type your question into the chat or raise your hand so that I can unmute you. I do see we have one question. So the question, nice talk, well done. Could you use data assimilation to improve the accuracy of the spatial variation of beta and delta?
That's a nice question. I haven't thought about it yet, but actually the question touched on the very important aspect will be the next step of this research. It's about this uncertainty or variation or like the accuracy of the spatial variations. Because we know that we find this spatial variation based on one of the algorithms, or this algorithm or this method may not be the best one. So I guess if we can find a more, but if we can find a model to explicitly model beta and delta instead of having to just change it against some other data, but fit the model on that, then it will be a good chance or good opportunity to apply the data assimilation. But then the question becomes yes or no, because as I mentioned before, the data assimilation relies one model and the satellite data, and the satellite data measures directly the soil moisture, but they are not measuring the beta and delta, because these are just some mathematical symbols we put into the model, and we don't have direct observation on that. So yeah, it's an interesting topic to touch on this model accuracy, but I'm a bit skeptical about how the data assimilation will fit on that. But yeah, thanks for the question. It's definitely a good touch on.
Awesome. Thanks so much, Jack. Fantastic presentation. We'll now move on to our next presenter this morning, and that is Mikayla Hyland-Wood. Mikayla, feel free to get started whenever you're ready.
Thank you very much. I'll just share my screen. Are you able to see that?
Yep, I think everyone can see that.
Fantastic. All right. Good morning, everyone. Thank you very much for joining me at my presentation this morning. My name is Mikayla Hyland-Wood, and I'm a third-year undergraduate at the ANU. Earlier this year, I completed an internship with ACUR on closing agricultural nutrient gaps with human waste streams. This project really came to life with the help of Dr. James Coulty, Dr. Ben McDonald, and Professor Jamie Pittock, so I'd like to give a big thank you to each of them. Declining soil fertility is one of the most serious problems the world faces. In many developing countries, such as Tonga, this is affecting both the yield and the quality of agricultural production. Generally, the nutrient gap found in these soils is a result of two factors: increased farming intensity and decreased crop variation. This progressively leaves less time for fallow, causing nutrient-deficient soils to grow nutrient-deficient foods. I hope that if my research isn't able to answer this big problem, it can at least spark some new questions. Human waste is the most underutilized and controversial waste stream available, earning it great potential for its incorporation into agriculture. This waste is often referred to as biosolids and is rich in nitrogen, phosphorus and potassium, NPK, and carbon because of our diverse omnivorous diets. These are the macronutrient building blocks that plants require to grow, and the composition of chemical fertilizers. Therefore, if human waste is processed correctly, it has the potential to act as a free and accessible alternative. My project uses the closed island system of Tongatapu, Tonga's largest island, which is home to 3/4 of its population and its capital, Nuku'alofa. The island is built from coral limestones covered with a thick ash deposit, which is historically rich in NPK and soil carbon. Over 2/3 of the land here is dedicated to agriculture, but it's mostly home gardens with some intensive production that grows taro, sweet potatoes, cassava, coconuts, banana, and coffee. The latter are cash crops, while the starchy vegetables are often consumed domestically. This island was chosen for its relatively clean waste stream. It has limited industrial and pharmaceutical sectors, and because they import $500,000 of chemical fertilizers annually. This money could be saved and repurposed if there was a cheaper domestic nutrient source. This diagram shows the general pathway of my research, where the management interventions align closely with the research questions. The barriers of management are touched upon in the limits and assumptions section, and the desired outcomes show the two main goals of the study: to improve soil nutrient levels and to work towards sustainable agriculture with economic benefits for the Pacific. To keep this really vast topic structured, I've formed a pinnacle research question and 3 sub-research questions. The sub-research questions involve: what is the nutrient breakdown of human waste streams? What waste conversion processes exist? And what cropping recommendations can be made to increase nutrient retention? The methods and the finding sections of my paper use these sub-research questions to retain consistency and clarity throughout. The current study was a literature review, where keywords were assigned as search terms and used to limit results in relevant databases. Appropriate values were found in existing literature and then scaled to Nuku'alofa's 23,000 person population. Biosolid and nutrient masses were measured in grams per capita per day, then multiplied by Nuku'alofa's population and by 0.8 to reflect an 80% efficiency factor. This factor is relatively arbitrary, but it reflects a non-perfect process. It can easily be scaled back to 100% for the model to be used in different regions. Using keywords in relevant databases continued for the remaining 2 sub-research questions. It became clear as my research went on that Pacific nutrient profile research was limited, and Tonga was no exception. Profiles from the United States were often used instead, which actually reflects the modern Tongan diet pretty well in terms of high sugar, high carbohydrate, and high preservative foods. Although this is a justified assumption, it doesn't reflect the true Tongan nutrient profile, which exposes a gap in the current body of knowledge. The second largest assumption is the high efficiency factor of 80%. We know that energy will be lost in collection, conversion, and application processes. Additionally, not all macronutrients are in a form that's accessible by plants, which lowers the usable levels further. An exact percentage for this couldn't be found in existing literature, but it should be investigated further. The study revealed a long list of findings, but most importantly is the glaring knowledge gap that exists with data specific to the Pacific Islands. Additionally, little longitudinal research questions the viability of long-term human waste-dependent agricultural systems. In sub-research question 1, research from the Southern Cross University and the University of Nebraska confirmed NPK content of US biosolids to be 4%, 2%, and 3% respectively. This equates to about 8,000 kilos of nitrogen, 4,000 kilos of phosphorus, and 5,000 kilos of potassium produced annually by the population of Nuku'alofa. That's about 17 tons of domestically available fertilizer alternative ready to provide a service. Question 2 aimed to find appropriate conversion processes. And there are 3 main techniques. Solar drying is known to be the lowest effort, but it's time consuming and has high macronutrient loss. The second technique is hydrothermal carbonization, or HTC, which uses heat and pressure to transform solid waste into hydrochar, retaining about 75% of initial carbon. This has carbon storage benefits and abates the issues of time consumption. Finally, pyrolysis thermally treats waste while retaining some nitrogen, but requires expensive and specialized equipment that's often inaccessible to developing nations. The optimal method is likely a balance of HTC and pyrolysis to maintain both carbon and NPK, but future field tests need to determine the feasibility of these techniques in Tonga. Finally, researching resilient cropping uncovered two goals: increase crop diversity through rotation, and split the fertilizer application to be once at planting and once about 3 months later to encourage soil retention. Perennial crops were also found to increase soil carbon and should be preferentially planted. These might include taro and coconut trees. One sustainable cropping rotation would be a legume cash crop rotation within a perennial intercropping system. These findings show that biosolids from Nuku'alofa have enough nutrients to sustainably farm about 50 to 200 hectares. This area was actually surprisingly small, which encouraged me to look at other co-benefits, including environmental restoration of waterways. Nonetheless, the food grown in these biosolid fertilizer fields has a number of pathways post-harvesting. The first would be domestic consumption, although a number of reviews that I covered suggested that this may not be openly accepted. Another option is agriculture for export, possibly to cruise ships. This route may help strike a balance between using an available but contested waste stream and not pushing the social boundaries within Tonga. It's clear here that the opportunities outweigh the risks in a clean waste system. And the lack of current research presents a range of knowledge gaps ripe for investigation. This highlights the role of ACUR, CSIRO, and other Australian research bodies. Finally, I'd just like to say what a great introduction to research this was, and I'm really looking forward to continuing my studies in sustainable agriculture next year in honours. Thank you very much, and I'm now happy to take any questions you might have.
Thanks very much, Mikayla. Outstanding presentation. There are now 5 minutes for Q&A. So firstly, judges, do you have any questions?
I'll kick off if you like. Well done, Mikayla. Really interesting presentation, and your passion for the topic is palpable. So looking forward to seeing what you do in your honours year and where it goes to from there. My question is around whether or not you had a look at the psychological I guess, barriers of humans, messing in that human waste area and getting over the yuck factor. I guess it's great in theory, but, to be able to roll this out and bring it into a mainstream kind of, I guess, method of fertilization and soil cultivation, you're going to have to get over that. So I'm wondering if that was explored at all or how that fits.
Yeah, absolutely. So the social side was definitely touched upon in the limits, and it was outside the scope of this research just because it's such a vast part of this, and it does need to be investigated further. I know that Southern Cross University is doing some really excellent studies in Pacific Islands testing social uptake and judging what those barriers to adoption might be. So some of them are religious, some of them are cultural. Some of them have to do with whether women are menstruating and whether that has any play into whether human waste should be used. So there's some really interesting work going on, but it was outside the scope of my research, but was touched upon as a limit. Yeah.
Thank you.
Did the other judge have any questions, or are we moving?
I'm just seeing the chat going off. I would be happy to defer to that. I mean, my question was largely the same as Melinda's, but perhaps with a little bit more specificity, or actually perhaps a little bit more global in its reach. You kind of mentioned there, Mikayla, the internal intake of Pacific Islands communities, etc., using this kind of method. I mean, I'd be more interested in kind of the global implications. I mean, could this be kind of mobilized for export, as you kind of mentioned? Perhaps one of the economies you could try and use this kind of stuff with is kind of cruise ships or something. But as you already have kind of pointed out, this is kind of beyond the scope of your research anyway.
If that's all from the judges, I think we'd like to move on to the questions from the chat. So the first question, Mikayla, is did you explore the benefits of removal of the waste in terms of reductions in freshwater contamination and near coastal water quality improvements?
Yeah, absolutely. So when I found out that the land that could be sustainably used was surprisingly small, I did look at a couple of co-benefits. So I'll just go back in my slides to the photo of Tongatapu here. And you can see, so this is Nuku'alofa just here in the middle of the photo. And this lagoon just below it, there's really big signs of eutrophication there, and that's largely because of human and animal waste being put into the waterways. This is what's traditionally done in many Pacific islands, is waste isn't kept on land, it's preferentially put into waterways. So there are really significant co-benefits of cleaning up these waterways, not only for stock, but also for humans, also for the growing of the tourism industry. You know, pristine lakes on Pacific Islands is a really big attraction. So there's a lot more here than just using the waste for farming, which really should be looked at in future studies as well.
Awesome. And the second question is, you mentioned that utilizing biosolids in agriculture would be beneficial in Tonga. You also mentioned it has co-benefits. Could you please elaborate on some of these co-benefits?
I think that may have been kind of part of that first question there.
Yeah, no worries. And then unfortunately, we've only got 10 seconds of Q&A left, but Steve had another question. I'm sure you can follow up with that after the event. But unfortunately, due to time constraints, we're going to have to move on. So thank you very much for the presentation, Mikayla. That was really interesting. And we'll move on to our third and final presenter, who is Tori-Lee Monk. So Tori, feel free to get started whenever you're ready.
Yep, thank you. I'll just share my screen. Okay. First of all, good morning, everyone, and I hope you enjoy. Okay. Relationships are important, especially in plant communities, as it turns out. Plant communities are built and upheld by a range of relationships and interactions, whether it is with the physical environment, say chemical compounds, signaling molecules, or other organisms. And when these relationships themselves interact, it is imperative to detrimental to plant communities, whether they exist in nature or commercial systems. The project I'll be discussing today explores the relationship soil fungi and secondary metabolites share with plant communities and what that might look like. Fungi are arguably the black backbone of plant communities. The functioning and composition of fungal communities have absolutely fascinating impacts on above ground vegetation, though the effects from fungi depend on their lifestyle or functional role in an ecosystem, such as symbionts, which have a mutualistic relationship with plants, often as growth promoters living within or around plant tissues. Then there are saprotrophs, which are more involved with nutrient cycling as free living decomposers. And of course, plant pathogens that cause disease. To analyze fungal communities in soil or root samples, we may use molecular techniques such as DNA sequencing. This enables us to identify species and their abundance. In looking at allelopathy now, allelopathy was popularized as this inhibitory plant-to-plant process. However, allelopathy may be best explained as both an inhibitory or stimulatory interference involving plants and microbes. This process occurs through the addition of secondary metabolites or allelochemicals into the environment from a donor organism. This can be from any number of plants or microbes. Perhaps the most interesting component is the complexity of roles fungi play in allelopathy. From being potential donators themselves, they can also be involved in physically spreading these secondary metabolites using their underground networks or vegetative body, therefore enhancing effects. Or, in a similar way, they could mediate the spread through decomposing the compounds in the soil. Quite fortunately for us, allelochemicals can be measured. We can use an approach called metabolomics, which is the detection and quantification of low-weight metabolites in a biological system. This enables us to get a snapshot of the metabolites present in a sample or the sample's metabolome. This is our site, located in the Bombala region of New South Wales. As you can see, the site is divided by 3 vegetation types: dense pine, medium nanna, and dense nanna as pictured. Our first observation is that the year-old pine co-occurring with nanna plants in these medium to dense nanna areas are dramatically smaller than the year-old pine in the dense pine area, as you see at the back. So you might be thinking, wow, is the native landscape fighting back? Surely nanna is directly inhibiting pine, right? However, the answer might be a bit more complicated than that. We found that even after burning away nanna plants, the pine growth did not recover. So maybe we should dig a little deeper. We took rhizospheric soil samples, the rhizosphere being the soil located around and most closely influenced by plant roots. The soil samples were then sent for DNA sequencing for fungal community analysis, as well as untargeted metabolomic analysis to see what potential allelochemicals we may find. Yeah. In our community analysis, we saw this highly significant relationship between community structure below ground and above ground, driven by vegetation types. We conducted a number of analyses, including a principal coordinate analysis picture on the bottom left here. This tells us about the maximum explainable variance, so we can assess how communities in each sample relate to each other through spread of variance. This plot in particular shows clustering by vegetation type, suggesting these communities are correlated and unique to whatever vegetation type they belong to. This was further supported by a permanova to determine statistical significance of this relationship. So you might think, well, what does this actually mean in a functional sense? Here on the bottom right is a stacked bar plot showing lifestyles. They will tell you just that. We can see that in dense pine plots, the communities are mostly made up of symbionts. Dense nanna and medium nanna, on the other hand, are structured quite differently, lacking that proportion of known symbionts. Moving on to metabolomics. We see this great similarity in trends, the highly significant relationship between metabolome and vegetation type this time. The principal component analysis shown on the bottom left shows incredible clustering of vegetation types. This is shown most clearly though on the right here, picturing a dendrogram, showing vegetation types contain distinct metabolomes. Okay. Now, when putting all this information together, we see fascinating symmetry, giving us a taste of how fungi and allelochemicals shape plant communities. It also gives an opportunity to ask questions from a variety of contexts, be it ecological, agricultural, or maybe just from someone with a great curiosity in what goes on below ground. And all in all, allelopathy is a complex process that is centered on plant microbe relationships and dynamics. And most certainly, fungi have an important role in this. Okay, thank you for listening, and I will now be taking questions.
Thanks very much for the presentation, Tori-Lee. So
you.
yes, we do now have 5 minutes for Q&A. So we start off with the judges. Do you both have any questions?
How large was the area you were looking at here, Tori-Lee, and how long did this research take to?
So I'll go back to the site picture. So it's actually really amazing how close together this little area is. Sorry, off the top of my head, I don't remember in terms of square meters it was, but they're very close. Like they share the same soil, but you could say like maybe a meter apart, really. Yes. And so it was the other part of that question.
I was just wondering how long.
How long it is.
Yeah.
So I handled the data part of this project. So from sequencing data and metabolomic output, which involved very intensive data analysis through RStudio and whatnot. So I originally started, say, in February, and I completed to a good portion within a few months, but still to this day, I still review this data to learn more and more about how it applies to literature because, like I said, that most is what's thought of in allelopathy is quite a narrow definition to how it was first originally defined, because obviously it's a lot easier to study things that pose a detrimental effect, and that's more demand. It does make it not important to look at, by and large, what that means.
Well done, Tori-Lee. I know it's always hard when you've been sitting there for a little while listening to some others, keep
Yes.
going, and you're
Yes.
waiting for your turn. So
you
kicked a goal. So well done.
Thank you.
My question is probably a little bit more hypothetical. Given the work that you've done, and you just touched on the fact that you're still kind of going through the data and learning more and more as you go. How do you think the work that you're doing or will continue to do will, I guess, challenge that existing definition of allelopathy? Have you had to think about that? How you might contribute in a literal sense?
Absolutely. So like I was saying, is that this applies to basically everyone. So ecologists would be like, wow, is this a potential for example, pine actually, there can be quite horrible effects after looking at land remediation and everything. So they may think, oh, well, what are the kind of, in a biochemical sense, that we could replicate and perhaps renew landscapes? That could be from the ecologist point of view. There's also fascinating just looking in advancing knowledge in an area in general. So I would be very interested in doing a literature review, for example, and looking at, well, is there more conditions at play here, such as nutrients and whatnot in the soil? So yeah, I hope that answered your question.
Thank you. Yeah, it does.
Thanks.
If there are no more questions from the judges, are there any questions from the general audience? And if so, please type your question either into the chat or raise your hand so that I can unmute you. So we'll just give that a few seconds just in case someone is typing. If not, Tori-Lee, I had a question. In terms of your PCA analysis, I obviously come from a sort of molecular biology sort of background, and I primarily use PCA analysis if I'm doing some form of RNA sequencing, and I use a different plot to present my DNA sequencing. Is there any reason why specifically you chose a PCA plot?
So in terms of for the DNA, I specifically chose a principal coordinate analysis, which was more looking at transforming the data firsthand. So it's more comparable because being biological systems, as I'm sure you're aware, so looking at different vegetation types, it's not always appropriate to directly compare. So I chose this in particular because trying to look at how these individual drivers through each plot, and if when you look at the maximum possible variance, if they could cluster and show similarity, which they did quite luckily, thankfully. So yeah, I hope that answers your question.
Yeah, no, absolutely. No, I thought it was a really cool way of presenting the data. So
thank you.
Really well done. And that does bring us to the end of the 5 minutes of the Q&A. And so altogether, that also brings us to the end of today's session. So thank you to our judges, Ben Eldridge and Melinda Tunbridge for today. And also a huge congratulations to our 3 student presenters. You all did a fantastic job. So this recording will be made available following the conference and when it concludes today. And I also hope to see you all in the remaining conference sessions throughout today. So thank you everyone and really well done.
Thanks very much, everyone.
Thank you very much.
Thank you.